Section 13.3, we're going to be learning about how different foreign molecules are able to elicit an immune response and what those foreign molecules really need to look like. Alright, so just some definitions. And really when we're looking at these definitions, we're in that stage 2 of the overall immune response, and that's going to be presentation of antigens. And at first you have to look, how does the antigen get into you? Alright, and then once the antigen gets past that first and second line of defense, what does it do and how does it do it? An antigen can also be called an immunogen. Alright, and an immunogen is simply going to be an antigen that stimulates an immune response. Not all antigens are actually going to stimulate an immune response, but hopefully they do. And if they are an immunogen, then yes, they do elicit an immune response. Now the immunogens actually need to meet different requirements for the immune system to start that immune response. And the first one is hopefully that it is foreign. So if you recall from section 2, when we were talking about the differentiation of, or the selection of the B cells and T cells, those that attached to a self-antigen were killed, right, or underwent apoptosis to die so that then they wouldn't expand into a subset of B cells or T cells that would attack the body. So it does have to be foreign. And there's also shape and size components that will indicate whether or not it will be a good immunogen or not. And accessibility, how accessible is that immunogen or antigen to the immune system itself? Is it part of a protein that is buried deep inside all of the other amino acids, and so it's not actually seen by the immune system because it's not on the surface of that protein? Alright, so that type of accessibility. And again, also, hopefully it's not a normal constituent of the body, so it's not self. Right? The immune system should be trying to avoid attacking its own self. So examples of immunogens can be the whole microbe or different parts of the microbes. Cells are substances that can arise from other humans, animals, plants, other types of molecules. So we may have talked about peanut allergies. There's gluten allergies. Right? So that's coming from plants. Other animals. That could be dander, horse dander, cat allergies, dog allergies, those type of things. And then other humans. All right, when you have a organ transplant, right, that organ is going to be matched to you so that that organ looks as close to you as possible, even though it's from another human being. Right? If that wasn't matched well, then your immune system is going to attack that new organ because it looks like it is self, or it looks like it's foreign. Right? The immune system thinks it's foreign, even though it came from another human. But that human is can be very different from you on a genetic level. All right, complex proteins and protein-containing molecules are generally more immunogenic than just repetitive polymers contained of a single type of unit because you just have more variety and there's a higher what's called affinity for the immune components to actually bind to and attach to that immunogen. So here you have some examples of good immunogens. Alright, in this case you have a large protein, but the proteins are the epitopes, the components that are on the cell surface. The blue and the yellow areas, those are going to be good immunogens, whereas something that may be down here in the very middle that's protected by all of this other by all these other amino acids may not be the best immunogen because they're hidden. All right, here you have foreign human or animal cells. So this is like a blood transfusion. Blood transfusion should be matched to you. Plant molecules. All right, we see a flower here. Some of us have hay fever. All right, that can be elicited by the particles of the plant, the pollen that is floating in the wind, and we breathe it and our bodies react to it. Microbial cells, different viruses. All right, those are all good immunogens. But here you have poor immunogens, and you just have this nice long, very bland, very boring polypeptide and a trisaccharide. There's not a whole lot of interest there. All right, so then your immune system actually isn't going to see it very well and respond to it. So now we are reviewing some of the terms that we saw in section 1 or 2. All right, epitope. Epitope is the portion of an antigenic molecule to which the lymphocyte responds. Right, it's the primary signal to the immune response, to the immune system, that the molecule is foreign. So here, all right, this entire protein here, that is the antigen, and then the individual areas right here, this yellow area, this blue area, this yellow area, those are actual epitopes. So you can see how a large antigen, one big protein, can have multiple epitopes on it. All right, now another word for epitope is a hapten, but the hapten in and of itself is not a good immunogen. All right, so the haptens are going to be epitopes that are small, and you've got just 2 pieces here in this yellow color. That's a poor antigen. However, if you take this poor immunogen and attach it to a carrier molecule, then this entire complex is a good immunogen and can elicit an immune response to the yellow portion here, even though if the yellow portion was by itself, it wouldn't induce the immune response. So the hapten itself is what causes the hapten itself is what the immune system responds to, but it does so after the carrier is attached to it. And this is good for vaccine purposes. All right, then there are alloantigens. All right, alloantigens are going to be antigens that come from the same species as a person or another animal or something. So it would be humans. And if your blood type isn't properly matched, then, and you get a blood transfusion, then your immune system is going to respond against those alloantigens, those antigens that are on the other human red blood cells that are circulating in you that are not typed properly. Those are going to be markers on the cell surface. So again, areas of the cell that, or the foreign object, foreign cell, that your immune system can see easily and respond to quickly. All right, and this is going to be the blood group. All right, so I mentioned the blood transfusions. And also what's called major histocompatibility profile. And that was in section 1 that I didn't talk about. But the major histocompatibility, also called MHC, is what would indicate a good organ match. All right, because you may be of a certain MHC type, whereas someone else might be of a certain MHC type, and it's different. So therefore, the two of you are not compatible. And that's really where organ matching is going to be based on, is this MHC profile. All right. Then, 2 other terms, superantigens and allergens. All right, superantigens are bacterial toxins. All right, and they are called superantigens because they are very potent when they act to stimulate the T cells. And the way that they do this is they actually attach to the T cell receptor in a different way instead of sitting down on the inside, so it's very specific. it is able to attach to the outside of the T cell receptor, and then it avoids that specificity. So if you had a T cell receptor that was specific for superantigen, right, the superantigen would fit down inside there just fine, and you would only activate very few T cells, the T cells that were specific for that superantigen. But by name, that superantigen is a superantigen, and so it binds outside of that T cell receptor. So then it doesn't have to stick with its specific receptor. It can bind to all of the T cells in your body at the same time if it wanted to. And by doing so, it activates the T cells at a rate 100 times greater than normal signals. And what it does is it then releases, those T cells release cytokines. And those cytokines cause a lot of issues such as shock, an increase in vascular permeability. So the fluid in your vasculature goes out into your tissues. You no longer have fluid in your vasculature, so your blood pressure drops and you go into shock. And it's a very serious scenario. And toxic shock syndrome is an example of this. And if a person develops toxic shock syndrome and isn't treated quickly with a variety of different medications, one to try to neutralize that superantigen, one to dampen the immune response that's going on, and one to counteract all of the different cytokines that are being produced and doing their own thing, but just at very extreme levels. All right, that person may die if all of those processes aren't followed properly and actually done very quickly once the disease is identified. All right, then allergens are antigens that elicit an allergic response, and we'll talk about that in Chapter 14, which will be the week after Thanksgiving. All right, then almost finished here with Section 3. Antigens have to be processed and presented to T cells in order for them to be activated. So, in most immune reactions, the antigen has to be digested. Once it gets picked up by a cell that has the receptor on it that can specifically bind to the antigen, pull it in through phagocytosis and digest it. And there are specific cells that are called antigen-presenting cells because they can pull in the foreign material, and inside its cell then will digest or break apart the antigen, and then will display different pieces of it back on its cell surface. So there are three types of antigen-presenting cells, sometimes just referred to as APCs. The macrophage and the dendritic cells, these are both part of the second line of defense. And then the B cells, utilizing the B cell receptor, and those are the third line. And these cells are able to present antigen through the MHC class II molecule. This is an example of a dendritic cell going through that process where you see over here you have the microbe, and this is a pink guy, so let's say it is a gram-negative organism. It's going to be pulled into the dendritic cell. All right, in this phagosome here, it's digested, and it is broken up, so you see these little pink pieces coming from this big pink piece. And there is this MHC class II receptor that combines with the broken up pieces of antigen. They come together in this nice complex. All right, so here you have the processed antigen, broken up little tiny piece, and so this would represent an epitope, and it's presented on the surface of the dendritic cell in conjunction with the MHC class II molecule, and the T helper cell comes along and you have this nice T cell receptor complex, the T cell receptor, the CD4 molecule, and the CD3 molecule.